Okay, welcome everyone. Our guests are coming into the room. It's great to see everyone here. My name is Scott, and on behalf of Sci-Train and all of our friends at Scifiniti Publishing, welcome to this exciting event tonight. This is going to be a great event. We've got great signups for tonight, and I'm excited to listen. This, I confess, is not my field, but I'm willing to listen and learn. We have a luminary in the field of cell therapy and engineering. This is Dr. Ken-ichiro Kamei is here from Scifiniti. Welcome. How are you?
Okay, great. Thank you. Thank you very much for your introduction. Yeah, thank you very much for coming to this my webinar today. So today I'd like to share with you about uh, okay, let me see here. Okay, can you see my slide?
Looks great. And while Dr. Kamei is getting set up here, let me just briefly introduce you to our guests today as they file into the room. So Dr. Kamei is the editor-in-chief of Cell Therapy and Engineering Connect. And I'll put some links in the chat window so you can explore the journal, download the brochure, and find out more about Dr. Kamei and the rest of the board for the journal. But Dr. Kamei, as you can see, he's the editor-in-chief, and he's very experienced in this field. Looking forward to hearing his talk. As you can see, he is professor of in fields of biology and bioengineering. And as I said, he is the editor-in-chief of the journal. So Dr. Kamei, looking forward to this talk today. I'll turn it over to you. And in the meantime, I'll share some links so people can explore more about your journal and about Scifiniti. So Dr. Kamei, over to you. Looking forward to your talk today.
Okay, thank you very much for your introduction. Yeah, so my name is Ken-ichiro Kamei, so just call me Ken. And so I work as the editor-in-chief of the Cell Therapy and Engineering Connect. So I'm really happy to share with you about our journal scope and also in addition to my research topics as well. So this webinar, I really focus on the stem cell innovation, how stem cell is really contributing for the advancement of the healthcare, and also the new medicine. So today, actually, there are several topics I'm going to introduce, but before to begin with, just briefly, I'd like to introduce myself. So here is my research career. I got a PhD degree from the Tokyo Institute of Technology, Tokyo Tech, in 2003. At that time, I working on electrochemistry and also the biosensor to try to make a new type of biosensor to detect the cellular responses. And then moving to the UCLA, United States, after doing the PhD. And then at the time, I tried to make a transgenic mouse model to study about the cyclooxygenase 2, which is a really important protein caused to the pain, neurodegenerative diseases, and also the cancer. So we established a new transgenic mouse model to study about COX2. So the research field at the time is molecular biology and molecular in vivo imaging. And then I moved to the other lab, still in the UCLA. At that time, I studied about the microfluidic platform for the application in stem cell biology and also the cancer research. So this is a research area is micro nano engineering and also the stem cell cancer biology. Yeah, then after I finished my PhD postdoc research, then I moved to the Kyoto University. And then I tried to open up the new research field, reverse bioengineering, to create a living system in vitro. So this is a combination of the stem cells engineering and also the material sciences. And then 3 years ago, 2022, so I moved to the New York University Abu Dhabi as an associate professor here, and still continuing on the reverse bioengineering to recreate the living systems in vitro and the stem cell engineering and also material science. So as you may see here, particularly in these recent years, I really focus on how we can use the stem cell for the advanced and complete drug discovery over human healthcare. So my focus is the cells. So cell is a building component of the human and also the animals. So this is really important. If we have a good cells, we can maintain our proper function. We can do the exercise. We can also study, and we can also have a good experience. But if we don't have a wrong cells, so it's we're gonna have a diseases, and then we need to have some new treatment to cure ourselves. So cell is really one of the key topics, and then cells is really important for our journal too. And this is my research motivation and also ultimate goal. So we have cells, and we know that cell is a major component of the human and animals. Then our ultimate goal is can we build up the human by using cells? So this is our ultimate goals. So not only making a human clone, but we try to miniaturize the human into the small devices in my research. So it's so-called the body on a chip to replicate human in a chip, to miniaturize the human in a chip. So by using such a miniaturized human, so we can study about the human physiological and also the pathological condition without the use of human. And then we try to understand such a human physiology and also the pathology. Actually, my motivation is coming from the get inspired by this his quotation, Professor Richard Feynman. He was a professor in Caltech, and also he obtained a Nobel Prize in physics 1965. So what he mentioned is that what I cannot create, I do not understand. So this is really engineering way to understand biological phenomenon, and then we try to utilize such an understanding for the engineer and also the therapy. So this is my motivation. And then back to these cells, and I like to ask you about one question. So you can see here, so we are human and we have many types of organ tissue. But do you know how many cells our body has, and also how many kind of cells we have? So please think about it a little bit. So actually, we have a 3.7 trillion cells with a 270 kind. Yeah, so we have a quite diverse of the cells with a huge number of the cells. If you study about the microbiome, actually that number of the microbiome is much bigger than the human cells. But I like to focus on the human cell in this presentation. Okay, so we have 3.7 trillion cells with 200 kinds. So this is a huge amount of cells. Yeah. Okay. But interestingly, such a variety of cells and such huge amount of cells, actually it's coming from the single cells. It's a fertilized egg. After fertilized egg and sperm together, and then it's going to be the embryo. So that embryo give you 3.7 trillion cells with a 270 kind. It's quite huge, capability to differentiate, and so they can proliferate. So this is quite interesting, and this is amazing cells. But in addition to the such a fertilized eggs, we also have another type of cell which can have a similar capability like a fertilized egg. So this is so-called stem cells. So some type of the stem cell can differentiate into the many kind of cell in our body, and then they can proliferate. Therefore, if we can use such a stem cell, that would be a great tool for the advancement of our healthcare and also the establishment of the new therapy. And actually, by using our current medicine, like current medicine, we still have many kind of diseases we cannot cure, like Alzheimer's disease, Parkinson, and so diabetes or liver cirrhosis. So those diseases we cannot cure. Also, sometimes we need to have a transplantation from the healthy donor to the patient, but it's also difficult to find out the good match donor to transplant. So to understand such a diseases and also to establish a new treatment or to curing method, so the cell therapy is one of the key to cure the diseases which we don't have any effective treatment until now. So that's why I like to introduce how we can utilize the stem cell for the cell therapy, and then what would be the new innovation associated with the cell therapy. So this is today's topics I like to introduce. So firstly, just briefly, I like to introduce about the stem cell history and also the cell therapy, and also the introduction of the stem cell. What is the stem cells? And then I like to introduce the case study of the vision and also the Parkinson disease. And not only such a case study, I like to also emphasize about the manufacturing of the stem cells. So this is really important part for engineer, how the engineer can contribute for the regenerative medicine. And then the last two topics, I'd like to briefly introduce about my research for the chip and also the artificial embryo. And then lastly, I'd like to spend some time to introduce our journal. Okay, so this is the history of the stem cells. Okay, starting from the 1988 until now. So in 1888, already at the time, so the stem cell concept, stem cell terminology already, coming up by the Dr. Boveri and also the Dr. Hoeker. So this is the very first terminology. And then later, there's also the first description about the hematopoietic progenitor cells. So this is the kind of the broad source of the plot. So this is already kind of announced. Yeah. And then so there's a first stem cell transplantation to treat with the radioactive exposure by the Dr. Math. Yeah. And also there's a further success of the allogeneic transplantation to cure the aplastic anemia. And the important timings here, so 1981. So this is the establishment of the first murine embryonic stem cells, so established by the Professor Evans, Kaufman, and also the Martins. So this is quite important to use the stem cells for understand the biological phenomenon. Yeah. And then the Dr. Kaplan also describing about the mesenchymal stem cells. And then in the first case of the human, Dr. James Thompson established human embryonic stem cells isolated from the embryo. And in 2007, Professor Yamanaka and also Dr. Takashi also established a human-induced pluripotent stem cells is similar to the embryonic stem cells. And then later on, so by using such a stem cells, so they try to utilize stem cells to cure the diseases which we don't have a treatment until now, like spinal cord injury and also the cardiac, severe heart diseases and so on. So at the early stage, so we just have only the concept, but afterwards, so the people try to find out stem cell is existing or not. And then after find out that such a stem cell really exists, and then we can culture them. And then we try to use such a stem cell for the clinical application. So nowadays, people really focusing on how we can use the stem cell for the clinical situation. So this is a graph to showing about the distribution of the pre-human pluripotent stem cell trial in the country. So this is including embryonic stem cell and also iPS cells. So as you may see here, the America and also Europe and also Asian country can really contribute to advancement of the stem cells based therapy. So as you may see here, so of course USA is one of the leading countries, and then followed by the China and also Japan, and many of the European countries also contributing for the clinical trial of the stem cell based therapy. And I don't go to the detail, but so this is an overview of the stem cell trial in the clinic. So as you might see here, there's some application of the where the stem cells are used, like ocular, central nervous system, cardiac muscle, immunoblood, and endocrine and stromal cells. So there's really a variety of the tissue and organs stem cell can be used to cure. And then so the number of the clinical trial is really increasing. Yeah, and interestingly, even so there are quite a different type of stem cell, but human ES cell is one of the still the major cell source to work with, because human ES cell has a little bit longer history compared with the iPS cells. So that's why people are still using human ES cells, but then there's some other types of iPS cells that can be used. And still the stem cell based therapy is new. So therefore, most of them are still at the phase 1 or phase 1 or 2 in the clinical trials. and then only limited number is goes to the phase. Yeah, so but this is still very important, to have such a progress. Yeah, and here you might see that the number of the patient which are already treated, like immunocells blood is a little bit easy to access, so that's why you can see that there's a higher number, and then stroma and so ocular also important. So, and then I like to move back to the fundamentals. Okay, so what is the stem cell? So what is the basis of the stem cells? So I don't go to detail, but I like to briefly mention about the definition of the stem cell. So definition is that, so they have two important characteristics. The one is a self-renewal. So this means that, okay, so there is a one stem cells, and then through the cell division, and then that one stem cell can give you the two daughter stem cells. So from the one cell, it's gonna be two stem cells we can obtain. So this is a self-renewal. So by doing this process, we can amplify the number of the stem cells as we desire. And also, stem cells need to be differentiated to the organs or tissue. So differentiation is also one of the very important characteristics. Okay, through the division, that this stem cell can divide it to generate two progenitor cells. It's bit one step closer to the differentiated cells. I mean, functional cells. So this is quite an important process. So self-renewal and also differentiation is a two important characteristics as a stem cell. Actually, in addition, so this is a unique feature. So stem cell also showing that the asymmetric division and then two generator, one stem cell and also the one progenitor cells as a daughter cell. So because stem cell also need to be maintained in our body, and then stem cells also need to differentiate to give you that differentiated cell. To maintain such a balance, to keep stem cell and to induce a differentiation, such an asymmetric division also quite important to maintain the stem cells and also to obtain the differentiated cells. So this is a quite unique feature of the stem cells. And then where you can find such a stem cells? Actually, you can find quite a variety of stem cells in the variety of the tissues, like a brain, a bone marrow, or kidney, egg. So there's so many kinds of cells you can find around that in the human body. But if you think about the application toward the stem cell-based therapy, actually there is a limited cell sources. The one is the pluripotent stem cell, embryonic stem cell, and also induced pluripotent stem cell. The other is mesenchymal stem cells. So they have several difference about the differentiation capability. But just I like to briefly introduce what is the embryonic stem cell, induced pluripotent stem cell, and the mesenchymal stem cells. Okay, so ES cell is obtained from the inner cell mass blastocyst, and then they can propagate it into cell culture. So we can proliferate many times by using the ES cells. And this ES cell has a differentiation capability to the ectoderm, mesoderm, and also endoderm. And then finally leads to the neural cells, adipose tissue, and also pancreatic tissue. Actually, there are many kinds of tissue cells we can obtain from these embryonic stem cells. And the iPS cell is obtained from the differentiated cells or somatic cells, I mean terminated differentiated cells, to reprogram toward a kind of stem cell stage by adding the SKM or SKM factors like OCT4, SOX2, KL4, and CMYC, and then reprogram it to make the pluripotent stage. So, and this iPS cell induced pluripotent stem cell is a similar characteristic with the embryonic stem cells. And so they can differentiate to all germ lineage of differentiation, and also we can obtain different kind of cell. That's why the stem cell has a potential for the regenerative medicine. And the mesenchymal stem cell is obtained from the mesoderm lineage, and then they can differentiate to the adipose tissues or bone and so on. But also, this mesenchymal stem cell has recently shown that the differentiation capability to the neural cell as well. So in these days, the mesenchymal stem cells also the quite important source for the regenerative medicine or cell-based therapy. So this is the two main source of the mesenchymal cells. And interestingly, in the case of the mesenchymal stem cells, so people also try to use this mesenchymal stem cell from the different cell sources, particularly for the biological waste. Actually, this paper is a review paper is published in our journal, Cell Engineering, Cell Therapy and Engineering Connect. This is a Dr. Holt. This is the leading author of this paper. Yeah, and so mesenchymal stem cell actually can be obtained from the variety of the other tissue, like a dental pulp, adipose tissue, and menstrual blood, as well as extra embryonic tissue, umbilical cord, placenta, and also amniotic fluid. Usually, those are the kind of biological waste, and then we don't use those waste material anymore. But in these days, we try to utilize the mesenchymal stem cell obtained from these other tissue or extra embryonic tissues because they have a quite good quality of the mesenchymal stem cell. And then after isolated these mesenchymal stem cells from the bio waste, and then taking also the effectivity and also the transport to the storage facility, then repurpose this mesenchymal stem cell for the regenerative medical application. So we don't want to use such a precious materials anymore. So we try to use that the mesenchymal stem cell from biological waste and then for the regenerative medicine. So this is another way to obtain the mesenchymal stem cells. And I briefly mentioned about so stem cell can be used for the regenerative medicine, but it's also quite important to use such a stem cell for the drug discovery. Okay, the one side, regenerative medicine is more like a direct way to cure the patient by using stem cell. But drug discovery is the indirect way to cure the patient. Because by using stem cell, we can also establish kind of disease model in combination with the gene editing. But then, so by using such a gene editing technology, we can add some mutation to cause the diseases. And then by using such a disease model, we can also identify the new drug, new treatment to stop the disease progress. So that's why stem cell and also gene editing is one of the way to cure, to establish a new disease model. So gene editing is also quite important to correct the gene mutation in the patient-derived iPS cell or patient-derived stem cells. So we also obtained the iPS cell or the mesenchymal stem cell from the patient. So therefore, we can also apply for the precision medicine or personal medicine to cure. So disease modeling is also leading to the drug discovery. So I don't go to detail about this one, but we can also utilize gene editing for the synthetic biology to produce some therapeutic agent released from the cells. So this is also other topics which are quite interesting about. Okay, so this is a kind of a stem cell topics. But we also need to think about stem cell is quite important, but how we can use the stem cell and then how we can differentiate to the targeted tissue. So this is a quite important things we need to consider. Because stem cell itself, actually it doesn't work. And we also need to have a functional tissue out of the stem cell. And then to induce such as differentiation from the stem cell to the targeted tissue, we also need to understand how the stem cells are regulated in our body. So this is a stem cell. And the stem cells are quite well orchestrated and well regulated in our body. So, and this is the environmental factor, as you may see here, like secreted factor, cellular component, like hypoxia, metabolism, physiological factor, extracellular matrix. Which is quite a huge number of the environmental factors can regulate the stem cell function and also differentiation process toward the functional tissue. So we really need to understand such a differentiation process by understanding of these environmental cues for the stem cells. But in a current culture setting for the stem cell, we could not control the such environmental cues as we desire. So therefore, we need to have some engineering tool to modulate the stem cell function and also the stem cell environmental cue. And then we are really working on the nano and the micro engineering. So I'd like to briefly kind of remind you about the size of the cells and also the size of the engineering tool. So x-axis is showing that the size, and then here you can find that the female, and the X then here in the center, you can find animal cells. So animal cells have around 2 to like 50 micrometer in diameter. So around this scale, I mean the micrometer scale, it's really important to utilize a microengineer to control or to manipulate extracellular microenvironment. Outside of the cellular microenvironment, we can control by using this microengineering technology, because this is very much of the cell side and also the microengineering tools. I think if you see the more smaller scales, so you can find out the mitochondria, protein, or lipid. You can also find a virus, but those mitochondria, protein, lipid is a cellular component. So, and if you can see this size, so this is a nanometer scale. So nanoengineering is really important to control such a cellular component outside and inside the cell. So in combination of the nano and microengineering, so we can really manipulate such a cellular environment. So we can apply the proper environmental factor to control the cell function and also the tissue formation. So I'm going to briefly introduce how we can utilize such a nano microengineering for the regenerative medicine. So I like to move into this the topics about regenerative medicine. So actually, regenerative medicine is really can cover actually regenerative medicine is a medical field focusing on the replacing, engineering, or regenerating human cellular tissues or want to restore or establish a normal function. So we need to recover such a normal function by using the regenerative medicine approach. And then, so this really get involved to cell therapy, tissue engineering, and gene editing, and also stem cell therapy. So, and then based on that, I like to introduce some case studies about this regenerative medicine, particularly already moved to the clinical trial. So one of the application is macular degeneration. So I mean the eye. So if you have a normal eye, and if you don't have any problem, so you can see the clear vision with a normal eye. So this doesn't have any problem. But if you have some problem in the eye, it's caused to the dry macular degeneration, and then you can see some weird spot in the eye, and then that weird spot is caused to the problem for the unusually seen fuzzy or destroyed vision, and you can see not clear and not structured vision through this damaged eye. And if you can also get some more severe condition like wet macular degeneration, such a red spot can be seen in your retina. And then that really causes the problem for your vision. Like a blind spot can appear in the center of the field of the vision. So it's really difficult to see through this because of this red dot. So we need to overcome this problem. But unfortunately, until now, in particular for the wet macular degeneration, there's some treatment, but that treatment can only slow down or delay the progression. So we cannot completely stop and we cannot completely recover of this wet macular degeneration yet. There's a dry macular degeneration, there's no treatment, only lifestyle modification. Like if you can take antioxidant, or if you already smoking quite a lot, and you need to stop the smoking, and also you need to also limit the alcohol uptake. So such a lifestyle modification, only such a lifestyle modification is getting not better, just getting slowed down. So this is only the way to kind of stops to manage such a macular degeneration. And then, so this is the first case, first in human clinical trials by using a human iPS cell-derived RPE, retinal pigment epithelium. So this is the retinal cells on the back side of the eyes. And then for the treatment of the age-related macular degeneration, AMD. So this is the very first clinical trial of the use of the human iPS cells. So, okay, so iPS cell we can obtain the human cell from the patient, and then we can reprogram it to generate the iPS cells. And then we can induce a differentiation to the RPE. And then these are many kind of things like a RBC or a suspended and then put into the patient and then to recover. So this is a process to use the iPS cell for curing the AMD. So this is a generation of and also transplantation of the human iPS cell derived RBC. So you can see, this kind of cells. Actually, there's so many cells is sitting together and then forming a seed. And then by using this cellular seed, and then they cut the desired size, and then transplant it into the human eye and to see what can happen. And they can see that after the 1 year, they are putting onto this damaged area, and then they are kind of confirming about they have a kind of good engraftment of the eye after the transplanted area. So they're also having the follow-up clinical trial of the transplanted iPS seed. So to make a long story short, vision function assessment confirmed that significant vision-related quality of life improvement and potential retinal sensitivity restoration in one patient with the dry AMD. So this is still ongoing. Number of the patients still small, but still we can have a kind of improvement of the vision and also the sensitivity restoration even in the dry AMD disease, which we don't have any good treatment now. So such iPSC showing that such a potential. So, but by using this RPC, actually this is not so stable. It's quite fragile because it's consistent with only the single layer of the cell sheet, so it's quite fragile. So we also need to have some good stable supporter to make it that RPC more stable. So therefore, people try to utilize some engineering approach to make a scaffold to stabilize RPC for the toward the clinical trial. So there's some PET membrane, some sheet, and also the nanofibers, and also some scaffold. So this is in combination of the material sciences and also the engineering tools to make such a seed, and then we can stabilize that RPC, and then more better engraftment and better transplantation efficiency, and also better restoring rate. So this is people really working on this kind of stuff. So in addition to the RPC for to improve the vision, so people also try to use such an iPS cell for the Parkinson's disease. So this is clinical trial is led by the Professor Jun Takahashi. So he firstly reprogrammed the cells from the patient, and then from the iPS cells, they can establish a neuron. And then that neuron contains the dopamine neuron, which is really important to improve the Parkinson's disease. So firstly, they put the first patient in October in 1918, and then 7th patient is 2021. So this process is still ongoing. But so this is a kind of clinical data after the transplantation of these new neurons in the patient. Yeah, phase 1 and 2 clinical study. So, but to make a long story short here, so this F18 dopa is one of the indicator to visualize active dopaminic neurons in our body, in brain. So, and also if that patient doesn't have a good activity of the dopaminic neuron, the Parkinson's disease, that signal is quite low. So pre-operation, so this patient could not have the activity of the dopamine neuron, but after the transplantation, 12 months and 24 months, they are showing, he is showing that the really good recovery of the dopaminic genic neuron activity. So this could be the improvement for the Parkinson's disease. So this is a really great sign to improve such a Parkinson's disease. Okay, so neurons and also the vision, so we can use such iPS cells. But but actually, if you want to cure the patient who has some diseases or injury in a large tissue, like a liver or heart, we also need to consider how we can obtain such a huge amount of tissue, amount of cells to cure the big, large tissue, like a liver or heart. So that could be the big issue. So if you consider about the lab scale and also for the cell culture by using such as small culture dishes. So each dishes we can obtain only 10 to 6 cells. But if you want to cure the patient, we need to have 10 to 9, 10 to 10 cells for each patient. So there's a big gap between the lab and also the patient. So we need to prepare the thousands of dishes for each patient. So that is phasing out problem. So therefore, manufacturing is quite important to advancement. To accomplish our goal for curing the patient with the stem cell. Starting from a very small vials, and then in the R&D scale, so we can use a flask, and then we can kind of mini size scaled up for the process development and the phase 1 and 2 clinical trials. We need to have still more relatively large scale. Yeah. And then we also need to have a kind of manufacturing and so on. So we need to have a really scaled up. But for the challenge of the scale of culture, we're going to have an issue about the low cell proliferation and also the massive cell death during such a scaling up process. So we need to overcome some problem. How we can improve that efficiency to increase the cell number, try with a minimum cell death. So this is a quite important point. And then in my research, we are focused on extracellular matrices because ECM has a quite important role as listed here, but it's also quite important for the self-renewal and differentiation of stem cell. And we also realize that such extracellular matrices in our body forming this kind of nanofiber. Okay, so this is a green color showing the extracellular matrices attached with the cells and then induce a self-renewal. And importantly, these extracellular matrices are forming the nanofiber structure, which usually we don't care in a normal cell culture. So we established nanofiber cellular matrices, and then this is applicable for the scaled up culture of the stem cell. So we so-called fiber-on-fiber matrices, which combination of the nano and the microfiber. And then we put the stem cell, and that stem cell can proliferate, and then they can kind of form such a colony. So we can quite increase the number of cells. And then so we have to try to use such a fiber matrices for the scale up culture. So this is more like a medium size of the culture condition. So this is a kind of conventional cell culture condition. We are using a starting condition and then put the cells then inside. So in the previous conventional method, so to increase the cell number, so they are leading to 17 folds in the mass. So it quite taking a longer time, but we can obtain a quite a limited cell number. And we also need to have a daily medium changing. And that medium is really cost to maintain the stem cell. But in the case of our method, so we can increase the cell number 40 to 50 fold in a week. It's quite a shorter time, but we can still increase quite cell number efficiently. And we can also minimize the number of the medium changing 2 times per week. So we can also reduce the cost. So we can obtain high cell number with high quality within a shorter period. So again, nanotechnology is also quite, can get involved to the cell-based therapy because, we can also contribute for the part of the manufacturing. And then this is really important to give us the better quality of the cell in a shorter time. So not only the manufacturing, but it's also quite important how we can fabricate such a tissue which we want to desire. Because each tissues organ, they are quite sophisticated, and then such a sophisticated structure need to have their proper function. But our culture system in the dish, we don't have such a good way to have such a culture system to make such an organ. So advanced 3D bioprinting is a really important way to make a relatively larger tissue by using cells. Cells is mixed in ink. So by using this ink and in combination with microfluidic devices, so we can generate quite sophisticated structure of the tissue which with a multiple cell type with a defined location. So by doing so, this is the micro muscles which compose of the muscle cells with endocellular cells. So this is forming the broad space network. So this is quite important to have such a sophisticated and physiologically relevant organ by using 3D printing for the stem cells. So this is also the published paper, Cell Therapy and Engineering Connect, and we published a review paper to summarize what is the ongoing process of 3D printing. So please take a look review paper that published in our journal. So I briefly mentioned about how stem cells can be used for the regenerative medicine and how engineering tool can contribute for the such a regenerative medicine. But from here, I'd like to mention about the disease modeling and also the drug discovery, because this is also one of the quite important application area of the stem cells. So particularly for the issue of the drug discovery here. So even if you want to get the one drug, so it takes over 10 years, and also it costs $1 billion US, but actually it's getting more and more. And mostly it's gonna be failure at the side effect. So the quite limited number of the drug candidate can go to the market. Yeah, so this is a kind of a hierarchical structure of the drug discovery and the development field. So from the basic research to the approved drug, so only the one. So this is showing the number of compounds. For the basic research, we can have over 20,000 compounds, and then we try to narrow down toward the preclinical research, clinical research, and also approved drug. But still, it takes time to also most of them get period, particularly for the clinical research and also preclinical research. So we need to facilitate this drug discovery process. But we also realize that at the preclinical trial, this is quite important, and also this is one of the big hurdles we need to overcome. Because preclinical tests, we are using animals, and by using such an experimental animals, we need to perform the variety of tests to confirm that this drug candidate. But animal tests could not recapitulate nor predict human responses for drug tested, and also the animal test has an ethics issue. And actually, very recently, FDA and the NIH announced that no more grant proposals solely rely on the animal testing. So for the use of the animals for the research, if you only focus on the animal testing, so FDA and NIH no more support such a research proposal. So we really need to have the in vitro human model as an alternative of the animal testing, including basic research, but also the drug discovery application. And then by using, it's also quite important, this model is quite important to mimic human disease by using human cell and animals. But again, animals, we don't want to use any animal. So we would have in vitro human model. So to mimic such a human to human by using a human cell and a chip, so organ chip or body on a chip is really important. And then nowadays, this is a quite hot topics we need to go through. And this organ chip or body on a chip, we can put many organs like the heart, bone, liver, or kidney into the devices, and then we can see the interaction between them. Or we can also see that how the disease progress through such an intertissue interaction. But anyway, the key component is a microfluidic technology in addition to the human stem cell. So by using this combination of the microfluidic technology and human stem cells, that organ chip or body on a chip can be applied for drug screening, toxicological testing, and also the disease modeling. And because the pluripotent stem cell is quite important technology for the disease modeling, because we can obtain the different kind of organ cells, which is really difficult. Previously, we cannot obtain. So that's why this is quite along with the discovery and also the development of the body chip technology. And so this is the scheme of the drug discovery and where that micro organ chip system can contribute. So from the in silico model, so this is the kind of funnel of this drug discovery to the patient to the market. So starting from this drug discovery part, we are doing an in silico model and also a little bit simpler model to find out what kind of drug candidate will be good toward the preclinical screening. And then afterward, like organoid, organ chip, or experimental animals is also quite important. Yeah. And then body on a chip and the patient on a chip is really important for the clinical study and then to the next. So on-chip system is really contribute at the stage of the clinical, preclinical, and also the clinical studies. So it cannot be really out there the such an animal model, but still we need to really focus on to advancement of on-chip system to replace such an animal. So in my lab, so we also working on a quite variety with the organ chip and body on chip system. So as you might see here, so we publish a quite variety with the organ chip system like eye, brain, heart, liver, intestine, and so on. And not only the single organ in a chip, but also we put two type of tissue into the one devices and to see how they are communicating each other and how they are important to contributing for this disease progress. So the one of topics we are working on is non-alcoholic fatty liver diseases. Yeah, so the alcoholic fatty liver disease is one of the quite important diseases need to be stopped. But until now, we don't have any good drug and treatment. The intestine absorb the fatty acid, and then that is resulting for the liver accumulation. So that the liver access seems quite important to understand. And actually, the gut and liver axis has a bidirectional interaction with each other. So we also need to investigate how they are communicating each other during the drug NAFLD process. So we established that this is modeled by using our body on a chip system. So this is a chip we established. And this pink color where we can culture cells. So this is made of the PDMS. And then they have a 6 culture chamber and the 3 set of a closed circulation loop, which we try to investigate the tissue interaction. Yeah, so to accomplish this such a introduction to these different type of the cells, then so this is to perform such a closed circulation loop, we can put valves and pump by using micro engineering technique. And then we are also actuating the micro valves and pump to have such a medium circulation chip. Yeah, so and then we can put the liver cell one side and the other side, gut cell the other side. And then we try to investigate their interaction with each other. So to make a long story short, so we can obtain the new result which cannot be obtained from this single cell culture. So gene expression analysis for the treated with fatty acid, and then we can investigate a new mechanism. So we can obtain a new pathway of the progression of this through the gene system. Okay, so time is a bit limited, so I like to moving forward discuss about the future regenerative medicine. So, okay, so I just briefly introduce about the cell transplantation, and also I'm also introducing about some organ chip system. So, for the advancement of the future regenerative medicine, there are many kinds of the topics need to be advanced, like DNA, RNA sequencing, gene editing, tissue engineering, organ chip, body on a chip, and 3D bioprinting. So those technological advancement is really allow us to understand the individual people and also what kind of treatment is really suitable for the individual person. And then by using organ chip, body on chip, and 3D printing and tissue engineering, so we can identify suitable structure of the organs and how we can implant that printed organ to the patient. So this is really important activity we need to consider. But in addition, we also need to have an AI, because by using the AI technology, we can really quickly obtain what kind of biological insight each patient has, and then we can really identify the effective treatment. So not only such a diagnostics tool, that AI also allows us to establish a suitable structure and a suitable function for each patient through this organ chip, the bioprinting, and also tissue engineering. So in the future, it's quite AI-guided precision medicine, regenerative medicine is really important to come in play. And this is not our future. Actually, it's really ongoing future. So we really need to have this issue. Okay, so lastly, I'd like to briefly introduce about the Scifiniti and also our journal. So Scifiniti is established in the 2023, and then it's based on the UAE. And then we also try to establish the kind of to help the global scientific community, including the MENA region. And then we also have a quite good number of the published journal, like cancer immunology, cell therapy, and so on. And myself work as the intern-in-chief of the Cell Therapy and the Engineering Connect. So this journal, our journal, Cell Therapy and Engineering Connect, can cover the quite a variety of the topics, but which relate to the cells, genetic engineering, stem cell technology, cell-based therapy, tissue engineering, immune engineering, neuroengineering, and so on. So this is really driving advancement in health and also the biotech. So this could be the potential kind of application of the cell therapy and also the engineering. Of course, improve the human health, including personal medicine, genetic engineering, and drug discovery is a really key component of the cell therapy and the engineering. But particularly for the engineering part, it's not only the human health, but also we can consider about the 3D bioprinted means. This is also kind of a new way to think about the consumption of the meat and also try to reduce the animal which can be used as a meat. So this is also the kind we can cover such a variety of topics in our journal, Cell Therapy and Engineering Connect. So therefore, we are really looking for your submission. And if you have any question, please contact with me and also the editorial office, whether that your manuscript is suitable or not, we can improve our. Okay, so this is my acknowledgement to my lab member in the Kyoto University, New York University, and Australian Pharmaceutical University. And lastly, I'd like to acknowledge to the Scifiniti to have such a great webinar and also Sci-Train as well. So they're also helping me to have such a webinar. Okay, thank you very much for your kind attention.
Thank you very much, Dr. Kamei. Absolutely fascinating talk. As someone very far outside this field, I'm astonished at the advancement of where the field is headed. We do have a couple of questions from our guests today. We've got a few minutes left. Would you be willing to field a few questions, Ken?
Sure.
Fantastic. We have a question from one of our guests, Rabia. Welcome, Rabia. Thank you for your question. I will try not to mangle this question again as a social sciences guy. The question is in the chat window, and Rabia asks, how is immunogenicity controlled in iPSC RPE transplantation? Autologous versus allogenic. I hope I didn't mangle that question too much. Dr. Kamei, does that question make sense to you?
Yes, definitely. Yeah, so of course, the people try to use the allogenic and also the, so those kind of iPS cells. Let me check more carefully. Where is that question? Yeah, autologous and also the allogenic. Yeah, of course, the best case of scenario is the autologous iPS cell transplantation. But if you do that, it's gonna be also costly. So at this moment, people also try to use not only the autologous, but also the allogenic. And then try to use a gene editing technology, try to minimize such a cause of the immunodejection. Because we also realize that some protein which differently expressed for the individual, but if we can eliminate that protein, and then that protein diminish with that protein also allow us to use the iPS cell for the different person. So this is also one of the important way to combine with gene editing technology to reduce the immunorejection. So yeah, again, so we're also using autologous, but also the allogenic is one of the key way to
Great. Thank you for your question, Ken, and thank you for your answer, Ken, and thank you for your question, Rabia. If you have any other questions, feel free to pop them into the chat. I do have a couple others here. General questions, audience members were asking if we will share the recording. We absolutely will share the recording tomorrow. You'll be able to review Dr. Kamei's full talk with the transcript. So we'll share that about 24 hours after the talk ends today. So check your email inbox. We'll send that recording out to you. And so thank you for your questions there. This is fascinating to me, Ken, because I work with authors here and overseas, especially here in Japan, who work in adjacent fields that are impacted by this field. So for example, I work with authors now who are doing CAR T studies, and it's just so fascinating. I have some questions related to this. Obviously, it's very important for different fields to work together. You've got collaborations between clinicians, you've got engineers, IT specialists, biologists, people such as yourself to advance this field. So related to this question, how does an open access journal like your journal, how does such an open access journal help these different fields of collaborators to collaborate and advance this field? What role does open access publishing play in helping advance this field?
Yeah, thank you very much. Such a great, quite important question, because if you don't have such an open access journal, and then people often like stick onto their own research field, and then they cannot explore to the new research field. So that's why if we can have such an open access journal, that the people who don't have expertise in this field, but still can easy to access. So this means that they have a relatively lower hurdle to access to the other research field. So actually, to have such an open access that really engaging to the different research field, and then to facilitate the collaboration through the like interdisciplinary research. So the open access is quite one of the key to enhance such a collaboration.
Absolutely. The collaboration is so important. Open access really helps to facilitate that. Thank you for your answer, Ken. And thanks for your questions in our audience. Some great comments coming in here. Comments from the audience saying, thank you very much, Dr. Kamei, for your talk. Brilliant talk, they said, and I absolutely agree. It was absolutely fascinating. Perhaps time for one more question. I noticed that we do have some early career researchers joining us tonight. I noticed from the signups that some are new to the field. So do you have any advice, Ken, for young researchers or clinicians who want to get more involved in your field? How would you advise them to get involved in this type of cell therapy and engineering? What skills are in demand? So we talked about the different collaborators and engineers and so on that need to work to advance this field. How would you advise a young, early career researcher who wants to get involved in this field?
Okay, thank you very much for this question. Yeah, so of course, young researcher, so they don't have so much experience yet, but it's really important to have a kind of open mind to communicate with other people. Yeah, because one person has a quite a limited capability to do one thing, but that's such a collaboration to we can do more, and also such a collaboration sometimes we also can generate a synergetic effect. One-to-one giving not only two, one-to-one giving like a 5 or 10. So such a collaboration is really important, and so this means that communication is quite important. So you need to be open mind to communicate with the people, and then just jump to this, to talk with someone else which who are you are interested in. So, and then you can have such a kind of feedback. So if you want to do something, and then you can get feedback from the expert, and then you can have a kind of facilitate your thinking and you improving your project. So jump to this different research topic, different research area, and communicate with the people is quite important. Sometimes I also attend to the conferences which I'm not familiar with. So as kind of outside of that research conference, but just jump into that conferences and then try to present. And then I also kind of explore the new network. So this kind of activities is quite important. Of course, for the earlier age, you need to you need to get your own skills, but later you also need to have such a communication skill to communicate with many people and to work together and then to accomplish a big goal like a cell therapy or something. Yeah, so I think such a teamwork is really necessary. So then you also need to have such a communication skill to work with.
That's a great answer. Thank you, Dr. Kamei, and thanks for your question to our guest member who asked about that. And good luck with your career. Hopefully, you'll be working with luminaries like Dr. Kamei in the future. So thanks for your question and thank you for your answer, Ken. And I think we're getting to the end of our time here. If you have a question that we're not able to answer today, would they be able to reach out to you, Ken, if they have questions about the field or about the journal?
Yeah, no problem. Yeah, please reach out to me, and also they reach out to the editorial office. So we are really happy to communicate with you.
Great, thank you, Ken. I've dropped another link to the journal there in the chat. So thanks very much to everyone for joining us. And thank you most of all to you, Dr. Kamei. Absolutely fascinating talk. I loved it. Again, it's not even my field, but I was just gripped and fascinated. So thank you very much for sharing your expertise with us. And thank you to all of our guests for joining. We'll be sending out the recording and a certificate tomorrow, 24 hours after this talk. And we look forward to having future talks with Dr. Kamei and hopefully some of the other journals at Scifiniti. So Ken, any final words before we finish today?
Okay, thank you very much for coming to the this webinar, and this is a really great chance for me, to introduce our journal. And I really also want to have such a feedback and also the your contribution. And again, I also open. So if you have any question, please reach out to me. And then I also not only the submission of the manuscript, but also the kind of future collaboration. This is really important. Yeah, there's another one question. It's coming.
Oh, there's one more question. Got time for one more, Ken? Maybe we do. From Rabia.
Yeah,
Yeah,
challenging the scale
Rabia
up.
asks, what are the main challenges in scaling up cell-based therapies from lab to clinic? Oh, that's a great question.
Yeah, exactly. So if you want to have a scale up, actually, liter scale is still okay. But if you want to increase the liter to like 10 liter or 100 liters, and then cell going to have a damage, and also even such a big tank, they have some inequality of the cellular quality. So some cell get dead, some cell is really good. So such a quality control of the cell is the one of the issues to encounter, and also we can really not have a kind of differentiation process afterwards. So such a quality control and also the differentiation process also going to be the one hard afterwards. But thank you very much for asking.
That's a great question. Thanks again, Rabia. And if there are any other questions that we couldn't get to, feel free to reach out to Dr. Kamei and team. And if you have questions about submitting to Cell Therapy or another Scifiniti journal, we've dropped links into the chat, so you can visit the Scifiniti team there. And thank you very much again, Ken, for lending your expertise to us today.
Thank you very much. Thank you very much for listening. And I really hope to communicate with you. Thank you very much.
Thanks, everyone. Have a great day.
Yeah, have a great day.